Thank you very much for such kind words of introduction, Brenda. So again, I'm Leonard Sachs from the Office of Medical Policy in the Center for Drug Evaluation and Research in the FDA, and I'm delighted to welcome you all to this 2023 Investigator Training course. At FDA, we've long recognized that clinical investigators and others involved in the trial enterprise are critical partners in development of medical products. We rely on you for all the clinical data we receive to support the approval of new medical products, and we are tremendously appreciative of the contribution you make to medical science and to the health of our society. Recognizing this, we've considered it really important to reach out to you over the years and to share our experiences as regulators in this interactive forum. As many of you know, we've been running this training course for well over a decade, and we've included lectures on non-clinical drug development, animal studies, toxicology, chemistry and manufacturing controls, clinical trials, statistical analysis, drug safety, ethics, and regulations. As we did last year, we decided to do an online course this year. These will be lectures tailored to a virtual environment. So we've limited this to approximately 2 4-hour sessions today and tomorrow. And the idea is to cover the most topical areas of interest to investigators. Today, we'll focus on selected topics related to the design of clinical trials. And tomorrow, we'll look at some of the non-clinical and the early clinical data that you may come across in investigator brochures. We will also discuss the responsibilities of investigators and investigator site inspections. By sharing our experiences inside FDA, we're hoping to provide you with an insight into FDA requirements that will give you as investigators greater confidence and expertise in conducting trials, and that it will improve the clinical trial enterprise in general. We're thrilled to see the enormous interest in the course with more than 10,000 registrants from around the world. I was fascinated to see all the comments in the chat coming in from every country around the globe. It reminds us that the development of new drugs and medical products is at the heart of medical communities everywhere on the planet. I also want to thank our speakers in advance. They're all colleagues of mine at FDA who have generously given their time to participate and prepare these presentations. I will apologize in advance for not being the most technologically gifted, but I hope we will be able to give you a satisfying experience as free of glitches as possible. And during the panel sessions, we hope you will send in your questions to make this as interactive as we can. So without further ado, I'd like to introduce our first speaker, Kim Smith. Kim is a dear colleague of mine, a nephrologist who's been at FDA for many years, and she's joined our faculty. She will be helping me with the moderation of some of the panels, and I'm going to ask her to kick off with a discussion of FDA Structure and Mandate. Thank you, Kim.
Thank you for that introduction. For the next 15 minutes, I'm going to provide a quick overview of FDA Structure and Mandate to provide some additional context for the rest of the course. So the objectives of my talk are to provide a brief history of the FDA, to describe the current mission of FDA, and to introduce FDA's legal and regulatory framework for drugs and biologics. So to start with the brief history of the FDA, I think it's important to recognize that the agency was built on a legacy of public health failures, so unsafe, ineffective, counterfeit, or adulterated drugs, as well as some unethical practices and fraud. The image on the right is an advertisement for Mrs. Winslow's soothing syrup, which was marketed in North America and the United Kingdom in the 19th and early 20th centuries as a cure-all medicine for fussy babies. But the primary ingredients were morphine and alcohol, and thousands of children are believed to have died from overdoses or from morphine addiction and withdrawal. The lower right is an image from the New York Times. This is on the Tuskegee syphilis study, which was started in 1932 by the U.S. Public Health Service working with the Tuskegee Institute. It was a study to record the natural history of syphilis. Participants were not consented and were told that they were being treated for bad blood. By 1943, penicillin was the treatment of choice for syphilis and was widely available, but the participants were never told about this or offered treatment. In 1972, the study was exposed when an article was published by the Associated Press, but unfortunately at that time, men in the study had been untreated for many years and their disease had progressed. To continue with our brief history, I'll provide an overview of just some key milestones in U.S. food and drug law. So starting with 1848, we had the passage of the Drug Importation Act, which required U.S. Customs Services to inspect entry ports for the entry of adulterated drugs from overseas. It's really one of the earliest attempts to regulate the U.S. Drug supply. Moving forward to the early 1900s, at that time, diphtheria patients were routinely treated with an antitoxin that was derived from the serum of horses. The horse serum was manufactured locally and there were no central or uniform controls. In 1901 in St. Louis, Missouri, the blood of a tetanus-infected horse was used to develop the antitoxin and was administered to 13 children who ended up dying of tetanus. This tragedy led to the 1902 passage of the Biologics Control Act, which allowed for the first controls over processes used to produce biological products, including vaccines, serum, and antitoxins. In 1906, we had passage of the Pure Food and Drug Act, which prohibited interstate commerce in misbranded and adulterated foods, drinks, and drugs. In 1912, we had passage of the Shirley Amendment. The Shirley Amendment prohibited labeling medicines with false therapeutic claims intended to defraud the purchaser, but it was a standard that was very difficult to prove. Moving forward to the late 1930s, in 1937, we had another tragedy. At the time, sulfanilamide was a drug that was used to treat streptococcal infections, and there was demand to provide it in a liquid form. So a company experimented and found that sulfanilamide dissolved in diethylene glycol, compounded the product and sent it all over the country. Unfortunately, diethylene glycol is antifreeze and is toxic. The product killed 107 people, many of whom were children, and really provided a dramatic example of the need to establish safety before marketing a product. This tragedy led to the passage of the 1938 Food, Drug, and Cosmetic Act. The act required that new drugs be shown to be safe before marketing and really started a whole new system of drug regulation, and it was the origins of much of what we do today. In 1944, we had the Public Health Service Act. The Public Health Service Act covered a broad spectrum of health concerns and included provisions related to the regulation of biological products. In 1951, we had the Durham-Humphrey Amendments. Durham-Humphrey Amendments defined the kinds of drugs that could not be used safely without medical supervision and restricted their sale to prescriptions by licensed practitioners. So this was our first foray into prescription products. We then have our next tragedy. Thalidomide in the 1950s and early 1960s was widely used to treat nausea in pregnant women. It became apparent in the 1960s that thalidomide was resulting in severe birth defects in thousands of children, limb deficiencies that you can see in the images on the screen. This was primarily occurring in Western Europe, and fortunately, the product had never been approved in the United States. Dr. Francis Kelsey, who was a FDA medical officer, had concerns about the amount of safety data that was available and had kept the drug off of the U.S. market. This example provided a lot of public support for stronger drug regulation and led to the 1962 passage of the Kefauver-Harris drug amendments. These amendments ensured drug efficacy and also enhanced safety controls. For the first time, drug manufacturers were required to prove to FDA that their product was effective before the product was marketed. In 1968, we had the Drug Efficacy Study Implementation, or DESI. Basically, many products had been approved between 1938 and 1962 on the basis of safety alone, and the agency had to generate a process to go back and review 4,000 plus drugs to determine whether they should stay on the market. The products that did stay on the market went through this DESI approval process. So fast forward to the FDA in 2023. The agency currently has approximately 18,000 employees. Many of them scientists, and the most recent numbers are 11,000 plus of these 18,000 employees are in science fields. The agency oversees the safety of more than $2.1 trillion worth of food, tobacco, and medical products, and this accounts for 15 cents of every dollar spent by U.S. consumers. The figure on the right shows that the largest program by far is the human drug program, followed by foods, and then devices and biologics. So I'm going to shift focus a bit to provide some information about the legal framework that guides the work that we do at FDA. Our work is grounded in statute with the Federal Food, Drug, and Cosmetic Act and Public Health Service Act providing our statutory authority for oversight of clinical investigations to evaluate safety and effectiveness. The laws are further explained through regulations that the agency issues. They can be found in the Code of Federal Regulations. This is how we implement the statutory authority over the conduct of clinical investigations. A few key sections. Title 21 is Food and Drugs, and Chapter 1 addresses the Food and Drug Administration. Subchapter A provides some general provisions. This is where you'll find information, for example, on informed consent and institutional review boards. Subchapter D addresses drugs for human use, and Subchapter F addresses biologics. FDA guidance is also issued regularly by the agency, and FDA guidance is intended to be advisory. It's intended to assist regulated entities in complying with the regulations and to help them to understand FDA's current thinking on a topic. There are numerous guidances, many of which are relevant to investigators, others might be more relevant to sponsors, and if you're interested in whether there's guidance on a particular topic, the link at the bottom provides the search location. So much of the agency's work revolves around review of submissions to certain types of applications. The first is the investigational new drug application, or IND, and then you have the new drug applications, NDA, or biologics license application, or BLA. And I'll talk a little bit more about each of these. So current federal law requires that a drug be the subject of an approved marketing application before it's distributed across state lines. Because a sponsor probably will want to ship the drug to clinical investigators in many states, it needs to seek an exemption from that legal requirement. And an IND is the means through which they technically obtain that exemption from the FDA, which allows them to ship the product across state lines. This, of course, allows them to initiate clinical studies in humans. There are three main types: investigator-initiated INDs, which I think would be the most common in most situations where you're studying a drug to meet an approval standard. There are also emergency use INDs as well as treatment INDs. There are two categories. There are commercial INDs. These are the ones that are initiated by a sponsor who intends to eventually seek some sort of a labeling change or approval of a product related to the studies that are conducted under that IND. For studies that are being conducted more for academic or research purposes, there are also research INDs. All INDs must include preclinical data, manufacturing information, as well as clinical protocols and information on investigators that will allow the agency to evaluate whether the study that's proposed is reasonably safe to proceed. Not all studies of drugs need to be conducted under IND. There are exemptions for specific types of studies. So first, the product has to be lawfully marketed in the United States already. This cannot be a new product that is not already on the market. The study cannot be intended to support a new indication or a significant labeling change for the drug. The study itself cannot involve a route of administration, a dose, a patient population, or some other factor that would significantly increase the risk to participants in the study. And the investigation needs to be conducted in compliance with other regulations for instance, institutional review board, informed consent, and promotion. When the agency reviews these INDs or reviews any submission to an IND at any point in a development program, one lever we have if we have concerns about the study is to issue a clinical hold. This is an order that's issued by the FDA to delay a proposed clinical investigation or to suspend an investigation that is already ongoing. When we issue a clinical hold, no new subjects can be recruited or given the drug, and the patients in the study need to be taken off therapy unless they're permitted to continue by FDA in the interest of public patient safety. For instance, if stopping the drug abruptly would be harmful, there can be a plan to manage that in particular situations. Not all clinical holds are complete. There are partial clinical holds. For instance, if you have enough data to support lower doses but not higher doses, there may be a clinical hold on administering higher doses. Or for instance, if you have enough data to support studies in adults but not children, the studies could be allowed to proceed in adults while there's a clinical hold on studies in children. So what are the grounds for a clinical hold? For phase 1, 2, and 3 studies, the agency could find that there is really unreasonable and significant risk of illness or injury to participants in the study. There may not be enough information to understand the risk, so there may just be more information needed before a particular study or part of a study could move forward. The investigator brochure, which is intended to provide information to investigators on what is currently known about the drug, may be misleading, erroneous, or incomplete and need to be corrected to make sure that information on the drug is being accurately and completely portrayed to investigators who are enrolling patients in the study. The clinical investigators may not be qualified to conduct the investigation. And then lastly, if there is a disease that is life-threatening and affects both genders, but one gender, often women, are being excluded, that could be another ground for a clinical hold. For phase 2 and 3 studies, all of the above apply, but in this case, the agency can also put a study on clinical hold if the protocol is believed to be deficient in design to meet the objectives of the study. Moving on, once you've conducted all of your studies under IND, it comes time for a marketing application. So as I mentioned, there are 2 types of marketing applications, depending on whether you're a drug or a biologic. There's a new drug application, which comes under the Food, Drug, and Cosmetic Act, or a biologics license application, which comes under the Public Health Service Act. Both of these provide for submission of information that's needed to support marketing approval of a new product, or to support provide additional information and claims for an already approved product. So the key decisions before us when we get either an NDA or a BLA are whether the drug is safe and effective in its proposed uses, and whether the benefit of the drugs outweigh the risks of the drug, whether the labeling is appropriate and what it should contain, so providers who read the label understand how to use the drug and who it would be beneficial in. And then finally, whether the manufacturing methods used to maintain the drug's quality are adequate and that the methods preserve the drug's identity, strength, quality, and purity. And so at the end of the day, if we find all of these to be true, we will approve a drug and it will be legally marketed at that point. I'll move on to the challenge question. So the first challenge question is for a drug that is lawfully marketed in the United States, an investigational new drug application is never required to conduct a clinical investigation using the drug. Is this true or false? So this is false. Not only does the drug have to be lawfully marketed in the US, but to be exempt from IND requirements, the study can't be intended to support a new indication or significant labeling change. It can't involve a route of administration, dose, patient population, et cetera, that would significantly increase risk. And the investigation needs to be conducted in compliance with other regulations, such as IRB and informed consent requirements. So challenge question number 2, which of the following is not a reason for a clinical hold? A, the investigator brochure is incomplete. B, there's insufficient information to assess risk. Or C, the investigation of a drug for disease that manifests in childhood excludes children. Or D, clinical investigators are not qualified, which is not a reason for a clinical hold. So C is not a reason for a clinical hold. Just to briefly summarize my talk, FDA has been working to promote and protect public health for over 100 years. It's one of our oldest consumer protection agencies. The laws, regulations, and guidance provide a framework for the development and approval of drug and biologics and really frames the work that we do day to day at FDA. INDs are the mechanism through which FDA evaluates whether clinical investigations are reasonably safe to proceed. And NDAs and BLAs are the mechanism for our oversight of the U.S. sale and marketing of drugs and biologics.
Thank you for the great presentation,
I'll be back
Captain
a little bit
Smith.
later
This is Ray Ford serving as your host for the remaining sessions. Moving into our next presentations, and the first one is on the basics of clinical trial design on design, population, intervention, and outcomes by Dr. Fred Senatore, lead physician, Division of Cardiology and Nephrology, Office of Cardiology, Hematology, Endocrinology, and Nephrology in the Office of New Drugs, CDER. Next, we'll learn about new trends in trial design and decentralized clinical trials, digital health technologies, and real-world evidence by both Dr. Leonard Sachs and Dr. John Concato, Associate Director of Real-World Medicine, Office of Medical Policy in CDER. This will lead us into our first Q&A panel. Please join me to welcome our next presenter in this session, Dr. Senatore.
Hi, my name is Fred Senatore. I'm a lead physician in the Division of Cardiology and Nephrology with the Food and Drug Administration, and today I'm going to talk about the basics of clinical trial design, specifically 4 elements: design, population, intervention, and outcome. Now, this talk is designed for clinical investigators. Many features of the topics in this presentation are usually a sponsor's responsibility. However, as a clinical investigator, you are responsible for enrolling subjects who have the disease of interest. You are responsible for not committing protocol violations. You are responsible for successfully migrating data to the sponsor or contract research organization. You're responsible for assuring subject safety, and you are responsible for assuring that each subject enrolled has been fully informed of the risks via a documented informed consent. Now, questions that come up should be asked by you, the investigator, if you feel the protocol is felt to be harmful to the subjects or felt not to be properly designed to evaluate a clinically meaningful primary efficacy endpoint. You are encouraged to question the sponsor, either individually or at the investigator meeting. Now, I'm going to talk about 4 learning objectives today, each with respect to the title of this talk. With regard to design, you will be able to appreciate what all the trial designs have in common. With regard to population, you will be able to identify the appropriate patient population for the hypothesis under investigation. For the intervention element, you will understand the various protocol designs and appreciate factors to consider in the design of clinical trials. And finally, for the outcome element, you will be able to describe primary and secondary. Now let's go to design. Pursuant to the Code of Federal Regulations 21 CFR 314.126, and I ask that you remember this particular statute, CFR 314.126, because it codifies the requirements for an adequate and well-controlled clinical trial. The trial must be adequate. That is, the trial must be designed to be able to discern a purported treatment effect based on a pre-specified criteria for success, and it must be well controlled. Usually a randomized trial is defined as a well-controlled trial that attenuates baseline imbalances of patient characteristics that could confound the results and jeopardize data interpretation. So the key message for the common trial features are that the data from the trial must be interpretable with a discernible treatment effect for drug approval. The trial design should include a pre-specified criteria for success. That is, the acceptability of a type 1 or false positive error, as well as a type 2 false negative error, the limits and boundaries where we might accept the type 1 and type 2 error. And as an investigator, you should speak up if you feel that the trial does not address a clinically meaningful issue or that the trial is not properly designed to meet the statutory requirements. Now let's go to the population. There are 3 types of populations. There's the intention to treat population, that is all randomized patients regardless of treatment. There's the modified intention to treat population, that is all randomized patients who took at least one dose of drug. And then there's a per protocol population, that is all randomized patients who followed the protocol exactly as written. Now, the ITT population is the preferred population that you investigate from the FDA perspective. The ITT analysis preserves the prognostic balance afforded by randomization, thereby reducing any risk of bias that may be introduced by comparing groups that differ in prognostic variables. The modified intention to treat population is tolerable. It's not preferred, but we normally accept it. The risk here is excluding subjects that could introduce bias and lead to misleading results. The per protocol population is seldom endorsed. By excluding subjects who deviated from the protocol, it introduces attrition bias, in which groups of subjects being compared no longer have similar characteristics. Now, with regard to the strategy for inclusion criteria, you would like to enroll subjects that are not too sick. You want to avoid enrolling subjects that are so sick that no matter what you do, it won't work. And then at the same time, you don't want to enroll subjects who are minimally sick. You would like to enrich the subject population with some risk factors to increase the chance of a patient experiencing an endpoint. So you want to optimize your selection strategy to not enroll subjects too sick or too well. But in the process of doing so, we need to be careful about regression to the mean. So a word about regression to the mean. One may select a subject experiencing either a bad day or a good day at baseline, that is, the tail ends of intra-subject variability. Such a subject may revert to their average level of illness during the trial. When that happens, the drug could be erroneously seen as effective if the subject had a bad day at baseline. Therefore, you're committing a type 1 error. On the other hand, a subject having a good day at baseline may empirically decrease the treatment effect of the drug. You're committing a type 2 error. Now, there's some remediation strategies to avoid regression to the mean. You should measure baseline values multiple times. Also, you should consider remote therapy monitoring, as it would apply to any eligibility criteria with intra-subject variability. Remote therapy monitoring may also capture extreme values beyond the tail ends that may not have been captured because of an exclusion criteria, thus avoiding potentially making a mistake that the treatment effect is being observed or an adverse effect is being observed beyond the intra-subject variability for the index disease presentation. The best defense here is a control group in a randomized trial. Now, a word about subgroups. The subgroup population is that which examines a particular characteristic like age, gender, race, ethnicity, region in the world, smokers, history of diabetes, or a history of myocardial infarction. The key questions that one should ask are the results the same for each subgroup? Are the results of the trial driven by only one subgroup? Is there a subgroup for which the drug is harmful? Now, there are two subgroup categories that you as the investigator should be cognizant of. The first subgroup category is one where the subjects are independent of any unique pathophysiology associated with or at increased risk for the index disease or primary efficacy endpoint. That means you're evaluating this subgroup for curiosity's sake. The second subgroup category are patients with unique pathophysiology that might matter to the treatment effect. These two groups should be treated separately when controlling for false positives. For the subgroup that might matter to the treatment effect, it is advised to have a pre-specified expected treatment effect prior to data unblinding. Now, with regard to subgroup analysis, a positive trial driven by one region, one country, one site, but negative everywhere else is still positive. A negative trial, but with a strongly positive subgroup, is still negative. Now, one must be careful about multiplicity when studying many subgroups for a possible claim. And the reason is the more tests you do, the greater the probability that you're going to be committing a type 1 error, a false positive. And this study was done 17 years ago, which demonstrated that fact. Now, with regard to the population element of this talk, the key messages here are data analysis should be conducted on the intention to treat or the modified intention to treat population. The intention to treat population should be selected to optimize the treatment effect of the intervention. One bit of caution here is that the label may be restricted to the ITT population if selection was based on a narrow severity spectrum rather than the broader population with various levels of index disease severity in the market. One must be cognizant about regression to the mean when enrolling subjects. One must also be cognizant about subgroup categories as pertains to a group that matters to the treatment effect. A positive subgroup in the setting of a negative trial does not constitute a basis for a claim, and testing too many subgroups will produce a false positive result. Subgroup analyses are therefore inconclusive. Now let's go to intervention protocol designs. Before we do that, I want to say a word about open or blind studies. An open label trial is one where everyone knows which patient is receiving what treatment. This may be a randomized control trial, or it may be uncontrolled. The pros of an open label trial is that it's easier to conduct, especially when blinding is not feasible. It's useful when comparing two approved drugs of similar characteristics, where each drug can be equally prescribed. It is useful in long-term extension trials to evaluate safety or persistence of effect. The cons of an open-label trial is intuitively obvious. It includes the possibility of bias. Patients or investigators may have a preconceived notion of the safety or efficacy of the drug that might impact their judgment. This is especially true when the endpoint is a soft one, like how do you feel, as opposed to a hard endpoint like death or myocardial infarction. Now, when it comes to blinded trials, this is a case where neither the investigator nor the patient has any knowledge about the treatment that is being given. And the key element of a blinded study is a randomized trial. Now, the pros of a blinded trial is that it attenuates potential bias in efficacy endpoint assessment, if that endpoint happens to be subjective, and attribution of adverse event to a drug. So, for example, if you're concerned about drug safety, knowing a subject is on the drug when experiencing an adverse event may result in assigning causality to the drug. So there's bias here. The cons of a blinded trial is that it's costly to perform. It's a much more complicated logistical regimen. You need randomization code generation. You need a blinded study drug kit. You need to design that. You need a distribution of the study drug kits, treatment assignment, and monitoring for study drug allocation assignment. So it's a much more complicated trial. Now, there are several blinded strategies that I think we should be aware of. There's the single-blinded trial, where the subject has no knowledge of what he or she is taking, but the investigator does. There's a double-blinded trial, where neither the investigator nor the subject know what is being administered. There's the double-blinded, double-dummy type of trial. This is a situation where it's similar to a double-blinded study, but each arm in the two-arm study, for example, has a different posology, pill versus intravenous or subcutaneous therapy. In this case, both dosage forms need to have a placebo mirror. Now, a word about randomization, which should be obvious to all of us. It attenuates influence of covariates on the primary efficacy endpoint. It's important to assess the effect of the intervention on the primary efficacy endpoint while minimizing the effect of confounders on the primary efficacy endpoint by a balanced distribution of confounders in the arms. It's as simple as that. You want to know the effect of the intervention itself on the primary endpoint without any other influence. So any other confounders are hopefully equally distributed amongst the arms in the randomization process. That is why the FDA always wants a randomization trial. And if you get a protocol from a sponsor where it's not a randomized trial, you might need to think twice about this. Now let's go into some randomized trial designs. What you see in front of you right now is a randomized parallel trial. Patients are going to be evaluated if they have stable disease. And if they do have stable disease, they'll be randomized to the active agent or placebo. That placebo may not necessarily be placebo. It could be an active comparator. This is the usual approach to evaluate superiority or non-inferiority. Screening for stable disease is a strategy to increase the probability of detecting a treatment effect. That is to say, enrolling very sick and unstable patients may cause the drug to appear ineffective. That is known as extreme enrichment. So you want stable patients in your trial, and you exclude patients who are not stable. Now, you could have another type of randomized parallel trial where there's a run-in period. The run-in period usually involves placebo or drug, and this usually evaluates compliance or toleration. And this also involves potential like drug discontinuation. That means if you're studying, for example, a hypertensive drug, and the patients they're already on a hypertensive drug, you wean them off and you put them on the test drug, and then if they tolerate it, you randomize them to continue drug versus placebo. Now, let's talk about a factorial trial. This is the case where you're going to compare drug A to drug B versus placebo versus the combination of A and B. The trial hypothesis in a factorial trial is usually that the drug combination is superior to the individual drug components. This is the case where you're trying to combine two pills into one, and this would involve a factorial trial. The crossover trial, patients are randomized to treatment A or treatment B. After a treatment period, there's a washout, and then the same patients are re-randomized. Those who took treatment A are now going to be given treatment B, and those who took treatment B are now going to be randomized to treatment A. The randomization is blinded, and none of the patients know if they got A or B initially, or B or A finally. This type of trial design is usually associated with the phase 1 pharmacokinetic trial of short-term design, approximately 2 weeks. It lowers the sample size. Crossover trials allow the response of a subject to treatment A to be contrasted with the same subject's response to treatment B. Removing patient variation in this way makes crossover trials potentially more efficient than similarly sized parallel group trials in which each subject is exposed to only one treatment. Now, here comes the placebo-controlled withdrawal trial. In this particular type of trial design, there is an open-label run-in period where everyone is given the drug, and everyone hopefully experiences a beneficial treatment effect. Then the patients are randomized to continue treatment or to placebo. So those patients on placebo will theoretically revert back to their baseline characteristics, and that difference between continued beneficial effect versus reversion to baseline characteristics would be your treatment effect. The primary efficacy endpoint is a relapse of symptoms following withdrawal of the drug. This trial design has been used for hypertension and pericarditis trials. In this particular trial design, one could measure persistence of effect and disease modification. So, key messages to the design aspect of our talk is that one must pick the right protocol design to fit the program hypothesis. The gold standard is a randomized double-blind clinical trial. It attenuates the influence of confounding covariates, and it minimizes potential for bias. Now, there are certain factors to consider in designing trials. The first one is the characteristics of the disease. The second one is your objective and hypothesis. The third is primary efficacy endpoint. The fourth is time frame and duration. This is important for you as an investigator. If you feel that the duration of treatment in the protocol is not aligned with what you believe to be the duration of treatment required for a treatment effect, you need to speak up about it. And there's also a cautionary situation here where the longer the trial, the greater the possibility of dropout. Therefore, as an investigator, you must have a subject retention plan as part of the trial design. Now, also as an investigator, you need to consider trial logistics. You need to streamline the execution of the trial and the acquisition of data from your site to the CRO to the sponsor's data bank. You must have a data migration plan in hand that is usually a sponsor's responsibility, where instructions should be clear to the investigators. If not, speak up. You need to take your data that you've acquired at your site and give it to the sponsor in a manner that does not jeopardize data integrity. There are ethical considerations that need to be made here. You have to ensure that patient safety is maintained. You need a safety monitoring plan, a trial stopping criteria. You need to ensure that patients are properly informed about the study, the risks, and the potential benefits. Without a safety monitoring plan, without stopping criteria, you need to inquire about it. Also, it's important to not jeopardize patient convenience. Don't create a hardship for the patients who are in your trial. Excess travel, for example, subjects bearing costs, especially for adverse events. You need to speak up if you don't think the protocol addresses ensuring patient convenience. There are statistical considerations that you need to be aware of as an investigator. The treatment effect should be discernible above subject variability on the feel, function, survive paradigm. The sample size should be aligned with the pre-specified criteria for success. Normally, we accept a 5% chance of committing a type 1 error, that is a false positive, and we also accept a 20% chance of committing a type 2 error, a false negative. That's where alpha of 0.05 comes in for committing a type 1 error, and beta is 0.2. That is the probability of committing a type 2 error. 1 minus beta is 0.8, and that is the power of the trial. If you're not clear on this when you read the protocol, speak up. Also, the primary efficacy endpoint should be clinically meaningful. The treatment effect should be large enough to have a net benefit for the patients. Now, the key messages here on factors to consider. When considering a protocol at your site, make sure the primary efficacy endpoint is one that you and enrolling patients deem important. If not, speak up. You need to ensure that the treatment and follow-up time frame is coincident with the treatment duration required to discern a clear treatment effect. You need to ensure the protocol has an adequate safety monitoring plan, a subject retention plan, and I'll say that again, a subject retention plan, because without subjects, and if they drop out, you don't have a trial, and you need a data migration plan. You need to ensure proper informed consent, hide nothing, and interact with your IRB. Now let's go to outcome. The outcome as defined by primary and secondary endpoints. The primary endpoint is defined as the specific clinical effect you wish to test. That is either a reduction in mortality, stroke, or heart attack. The statistical analysis plan and the sample size focuses on obtaining interpretable results and being able to draw conclusions regarding the primary endpoint. The primary endpoint can be a single variable, mortality for example, or the primary efficacy endpoint can also be a composite of several components. It's important to assure that each component of the proposed composite primary efficacy endpoint have similar degrees of clinical importance, that is to say, death, stroke, and heart attack, compared to death, stroke, and hair loss. If the second set of endpoints was the composite endpoint, and the results were positive, driven by hair loss, then the trial would be positive, but for a clinically meaningless reason. Secondary endpoints are of interest, but do not constitute the basis of a trial design. Now, sometimes a clinically meaningful endpoint, like mortality, is designated as a secondary endpoint, and the trial is powered for it. And the primary endpoint is excessively powered. That happens sometimes. Mention of the secondary endpoints in a label depends on whether they were part of a pre-specified alpha conservation strategy. This will be the subject of another talk about how to preserve alpha. Sometimes a mortality benefit, even outside of the alpha conservation strategy, may provide sufficient clinical meaningfulness to warrant a label claim. Now, I'm going to say something about the leeways of Phase 2 and the rigor of Phase 3. Phase 2 is a flexible type of study. The primary endpoint can be biomarker or a putative surrogate, and usually Phase 2 is to inform on the safety profile and the posology to Phase 3. Now, Phase 3, on the other hand, the primary endpoint must be clinically meaningful within the field function survive paradigm. So if you get a protocol from a sponsor, and it's a Phase 3 protocol, and the primary endpoint is a biomarker, like, for example, NT-proBNP as a primary endpoint, there's something wrong with that, and you need to speak up. So key messages for the outcome element of this talk is that the primary efficacy endpoint leading to a claim should be clinically meaningful within the field function survive paradigm. When analyzing the results of the trial, evaluating a composite endpoint of several components, consider the impact of individual components going in different directions. So if you have death, MI, and stroke, MI, myocardial infarction, and the study was positive because you prevented a lot of MI, stroke was neutral, but there was more deaths in the drug arm versus the placebo, that becomes a review issue. And when you look at unblinded data towards the end, this becomes an issue of whether or not the drug can be approved. So in conclusion and in summary, from the design element, as an investigator and clinical trialist, you are responsible to ensure the trial satisfies the statutory requirements pursuant to the Code of Federal Regulations 312.126. You are responsible to maintain knowledge on the regulatory requirements to safeguard your patients when enrolling them in a clinical trial. With regard to the population element, do not enroll very sick subjects during enrichment where the drug may not be effective. On the other hand, do not enroll a minimally sick ITT population where the drug may not have a discernible treatment effect. Be cognizant about regression to the mean when enrolling subjects, and use your clinical knowledge as an investigator if you think the protocol eligibility criteria are enrolling subjects at the tail ends of variability at baseline, thus potentially impacting the results. From the intervention point of this talk, focus on patient safety by ensuring the existence of and adherence to an adequate safety monitoring plan. Understand the data migration plan and the subject retention plan and adhere to it to minimize missing or corrupt data. Keep control of your site. Maintain vigilance over the research team and correct any issues in an expeditious way. With regard to the outcome, if you, as an investigator, happen to serve on a steering committee, you should ensure the primary efficacy endpoint is clinically meaningful. That means based on field function survival, and discernible by the subject above the normal variability of disease presentation. As an investigator, either on a steering committee or simply being an investigator, if you feel these things are not the case, speak up. So in conclusion, ask the sponsor if you have any questions. That's the most important thing. You, the investigator, have a significant say in the trial design. The investigator meetings are designed to ensure the protocol is consistent with the clinical practice paradigm. You, the investigator, are responsible for your patients enrolled in the trial. That is safety, informed consent, trial with a clinically meaningful outcome, and data management at your site. You, the investigator, are ultimately changing the practice paradigm for the better by bringing into the market a drug that you will have contributed to in demonstrating a beneficial treatment effect. And I want to thank you for your service to our society. This is an important role that you are taking as a clinical investigator. Now, I do have a couple of challenge questions. The first one, if a sponsor wishes to include the primary and series of secondary efficacy endpoints in the label, it is incumbent on the sponsor to develop a hierarchical analysis plan for testing each endpoint by pre-specified order in order to avoid committing a type 1 error. I'll repeat this. If a sponsor wishes to include the primary and a series of secondary efficacy endpoints in the label, it is incumbent on the sponsor to develop a hierarchical analysis plan for testing each endpoint by pre-specified order to avoid committing a type 1 error. Is that true or false? I'll give you 10 seconds. The answer is true. And here's my second question. Which of the following are true? A global trial is considered positive if the entire population, that is the intention to treat population, met the pre-specified criteria for success, even if subjects in the United States did not show a benefit. B, attempts to determine specific subgroups for which the drug will benefit the most in order to better guide healthcare practitioners may increase the probability of committing a type 1 error. C, a positive trial that is driven by only one region of the world, that is to say Russia and Ukraine, which is normally the case, but negative everywhere else, disqualifies that trial from having met the evidentiary standard. D, A, B, and C are correct. E, A and B are correct. Or F, B and C are correct. I'll give you 15 seconds. The answer is A and B are correct. The reason why C is not correct, a positive trial that is driven by only one region of the world is still a positive study. So that concludes my challenge questions, and I want to thank you very much for listening to this talk, and we'll be available for questions should you have any. And good luck with the rest of the talk, and have a wonderful day.
We're going to spend the next half hour discussing some innovative trial approaches, including digital health technologies and decentralized clinical trials. I think we all recognize that there's been significant progress in biosensor development and in communication technologies, and the opportunities for remote collection of data are quite remarkable. These technologies, as we know, are playing an ever-increasing role in clinical care, and digital health technologies and electronic communication platforms can obviously perform a significant role in the evaluation of medical products. Why bother with these technologies and approaches? This is a child with Duchenne's muscular dystrophy. As many of you know, Duchenne's is an inherited condition in young boys. It results in progressive muscle weakness, eventually tragically resulting in death, usually from respiratory failure when patients reach their teens. And this boy is participating in a clinical trial. In order to get to his trial visits, he has to be taken out of school. His mother has to take time off work. A flight has to be booked. They have to get onto the flight, transport the wheelchair, get into a hotel, and eventually get to the trial site. A very arduous and disruptive process. When they get to the trial site, this is what gets done. This is the 6-minute walk test, and the child is supervised, and the child is told to walk as far as he can for 6 minutes, and the distance is measured. It certainly leaves us scratching our heads. This is a picture of an accelerometer. These are tiny devices. Typically 3 of them are used together, in mutually perpendicular directions, and they provide a very good recording of all sorts of movements, including, of course, steps. Accelerometers are versatile tools for Duchenne's and for many other diseases. Accelerometers are used all over. We have them in our cell phones, our smartwatches, Fitbits, and so on. They can be very helpful in measuring steps or other activities taken by a Duchenne's patient with no need to depend on snapshot measurements or clumsy tests like the 6-minute walk distance. They can measure novel aspects of functions such as stride velocity, and in Duchenne's, the 95th percentile of stride velocity has been considered a promising measure to assess drug efficacy. And finally, comparisons with baseline measurements can be made. They capture objective data on functionality, which has traditionally been challenging in clinical trials. They may be useful in neuromuscular and cardiorespiratory diseases, muscular dystrophy, Parkinson's disease, heart failure, chronic obstructive pulmonary disease, and pulmonary hypertension. And they have a potential role in neuropsychiatric diseases, depression, ADHD, and schizophrenia, which are difficult to measure using current methods. And here are a couple of publications on the use of accelerometry. The first is by the European Medicines Agency, talking about quantification of stride velocity as an endpoint in studies in ambulatory Duchenne's muscular dystrophy. And the other one's a report in the New York Times where they report wearables track Parkinson's disease better than human observation. As far as biosensors go, they measure clinical features, and these may either be discrete events like a step, a breath, a cough, a pulse beat, a seizure, tremor, or forced expiratory volume, or they may be continuous readings like we get from a continuous glucose monitor, glucose readings, pulse oximetry, temperature readings, electrocardiograms, and blood pressure. Biosensors begin as a transducer, an electronic device that transforms a physical finding like acceleration or temperature or pressure into an electrical output. As you can see, there are a number of different types of transducers that we see in use: galvanometers, accelerometers, photoelectric cells, electrochemical sensors, and thermocouples all detect different physical properties. The output, as I mentioned, is some electric output, which might be either voltage, current, or impedance. And if these are intended to measure a clinical feature, for example, things like heart rhythm, walking, scratching, tremor, or blood oxygen saturation, etc., an algorithm is needed to process the data. Sometimes the algorithm includes calibration curves, which are needed in electrochemical sensors. And the result of the sensor and the algorithm is a clinical digital health technology. As you can see there, a cardiac monitor, an activity tracker, pulse oximeter, continuous glucose monitor, and a digital thermometer. Can we rely on the DHT? Verification is generally a process that's conducted in a lab, and its intent is to determine how accurate the DHT is in measuring the targeted physical feature, for example, acceleration or temperature. Is it reproducible in different environments, different temperatures, different humidity, and so on? Validation is usually done in the field, and the intent of validation is to show how suitable the DHT is for its intended use. Are the data recorded by the digital health technology in the patients the same as the data we would report if we were looking at the patient? For example, steps in a patient with Duchenne's muscular dystrophy, or steps in a patient shuffling with Parkinson's disease. Is the result affected by how the patient wears or uses the DHT? And are there other things that a patient might do that would be misinterpreted by the DHT? For example, tapping a foot or riding a bike. The suitability of DHTs for use in a clinical trial is obviously very important. Are they ugly or elegant? Are they easy to put on? Are they easy to operate? Are they comfortable to wear for the required duration of the study? Is the battery life convenient? Can data be synced? And can patients bring their own devices? I wanted to end this discussion on digital health technologies by mentioning that mobile applications are also considered DHTs. And here you can see a couple of examples: a patient-reported outcome on a cell phone camera that's used to capture a lesion, a coordination test in Parkinson's disease on the top right. This test has 2 buttons on the cell phone, and the patient's required to alternatively tap each of the buttons to test their coordination. And finally, things like visual tests on a cell phone. What about endpoints based on DHT measurements? Justification of the endpoint is important as a clinically meaningful measure of drug effect. When we think of the endpoint, it has various components. The first is what is being measured, for example, steps. What is the time window of observation? Let's assume this is 4 weeks. And what is the formula for the response in each patient, which in this case may be changed from week 1 to week 4 in the average daily step count. Another question is whether the endpoint is a clinically meaningful measurement of drug effect. And this can be established by comparison with existing benchmarks of performance, for example, things like the Unified Parkinson's Disease Rating Scale or other patient-reported outcomes, the 6-minute walk distance. It would also be very important to get input from patients, from caregivers, professional societies, disease experts, and regulators regarding the meaningfulness of the endpoint. I put the slide in just to remind ourselves of the differences between validation and verification versus justification of an endpoint. I think it's important to remember that validation and verification are technological assessments. They address how well the technology measures the clinical feature of interest. On the other hand, justification of an endpoint or a clinical outcome assessment is a clinical issue. It addresses how well the clinical feature of interest represents a meaningful response to treatment, and it has nothing to do with the DHT or with the method of measurement. Where are digital health technologies likely to be of use in drug development? And what I've done here is summarize the endpoints that were used in registrational trials between 2015 and 2020. You can see that there were 218 new drug applications during this period, and about 30% of them, shown in beige, relied on a biomarker, for example, a chemistry measurement like hemoglobin A1C, pregnancy test in contraceptives or GFR, hematology tests, pathology tests, and microbiology tests. About 22% relied on imaging. A lot of these were cancer trials where imaging was part of progression-free survival assessments, but there were other applications that looked at bone mineral density or vertebral fractures and spleen volume. About 7% focused on physiological or functional measurements. These included things like the 6-minute walk distance, normal sinus rhythm, forced expiratory volume, or sleep studies. 22% relied on clinical events or clinical signs, and these were generally prophylactic studies. There were studies designed to prevent things like death, hospitalization, major adverse cardiac events, or multiple sclerosis relapse. And finally, 32% relied on clinician-reported outcomes or patient-reported outcomes. And these were things like the Hamilton Depression Rating Scale, rheumatology scales, psoriasis severity indices, seizures, sleep, prostate symptoms, and other scores. So there are many types of endpoints where mobile technology tools may play a role. First of all, as we saw, clinical lab measurements, continuous glucose monitoring, and pulse oximetry are some of the obvious ones. Physiological measurements like heart rate and rhythm, breathing and lung function, seizures, syncope, temperature, and weight. And finally, performance assays, things like stamina, strength, coordination, abnormal movements, sleep, and cognition. Advantages of these type of continuous readings by biosensors are richer data instead of snapshot data. For example, we can get things like the average number of steps a patient takes a day versus the 6-minute walk distance. continuous glucose monitoring versus just a single hemoglobin A1C and diabetes. They give us the ability to detect rare events like arrhythmias, seizures, or apneic spells. We can get data from patients who cannot report, for example, scratching in infants with atopic dermatitis, or sleep patterns in patients with dementia. We can get dose response information, for example, the on-off effects of treatment in Parkinson's disease. And we can also make new types of measurements that weren't possible before. For example, accelerometry measurements of gait stability that may predict falls in patients with movement disorders, measurements of coughing, sneezing, and tremor, and behavior patterns in patients with dementia or depression. DHTs can be used for things other than endpoints. They can be used for enrollment, screening, and enrichment, helping qualify baseline disease severity and functional status. They can be used to monitor certain aspects of safety, for example, identification of rare adverse events, and they can also provide investigators with real-time access to patient safety data. They allow visualization of dose responses over the dosing interval. And finally, as far as endpoints go, their use would be most compelling in superiority studies, which show that one arm is better than another. Non-inferiority studies may be challenging to interpret. If the DHT is not working properly, it may give us the false impression that both arms are equally effective. What is our regulatory position on digital health technologies? We don't have regulations that directly address the use of DHTs in clinical trials. DHTs in clinical trials generally do not need to be approved or cleared by FDA for marketing unless they are medical devices. Our regulatory standard are the qualities of the data from a DHT adequate to provide substantial evidence of effectiveness? And finally, are the data attributable to the patient? Are there processes in place to ensure the data integrity, data security in transit and during storage? And you can refer to our guidance on the use of electronic health records and electronic signatures in clinical investigations listed below for more details on Part 11 as it applies to data integrity. Now let's turn our attention to decentralized clinical trials. Decentralized clinical trials are really a package of strategies. These are trials where some or all of the trial-related activities take place remote from the investigator at a place or location that's convenient for patients and that do not require visits to research site. The strategies include video and telemedicine visits, the use of digital health technologies, direct distribution of the product to patients' homes, the use of electronic informed consent so that patients can sign their consent at home, the use of home visits by either clinical staff or local healthcare providers, and of course the use of local healthcare providers themselves and facilities to provide some of the data used in the clinical trial. Why are regulators interested in decentralized clinical trials? Well, firstly, accessibility. This would be very important in patients with rare diseases who widely spread across the country or across the world. Second of all, for patients with mobility or cognitive challenges who have difficulty getting to trial sites. And thirdly, decentralized trials may improve the diversity of participants. For example, those who have socioeconomic challenges and cannot afford to take time off work where they're paid by the hour, cannot afford to employ child support while they're away. Obvious advantages for patient convenience, not having to get to trial sites. And then there are other efficiencies. For example, decentralized trials may avoid the costs associated with travel. They may reduce the need for physical facilities where trial-related activities are conducted. And they may also take advantage of qualified community providers who are doing a good job in clinical practice and may provide useful trial-related services as well. And then finally, our experience with COVID-19 has shown that these decentralized approaches are very important to prevent the spread of contagious diseases. Some concerns about remote data acquisition. It's important to ensure that there's oversight of trials. as trial-related activities get delegated to local healthcare providers and others more remote from the investigator, oversight has to be carefully planned. There would be potential concerns about device failure. Patient safety is obviously a primary concern. If there's any risk to the patient's health by not being close to the investigator site and not having access to other care, then that would not be a suitable environment for a decentralized trial. Patient retention has been a potential problem. Some patients just prefer the regular face-to-face visits that they have with their providers, and they may be less interested in engaging through remote technologies. Data privacy is also something that has to be ensured in these decentralized environments. Decentralized clinical trial procedures are not new. Patient diaries and interactive voice response systems have been used for years. Telephone follow-up has been common, particularly long-term follow-up in trials. In 2016, we published a guidance on electronic informed consent, which allowed consent to be done from patients' homes. In 2014, the first entirely web-based trial was conducted under IND. This was a trial looking at tolterodine for patients with overactive bladder. About 5,157 patients were registered for the trial. There were a lot of arduous procedures, including micturition diaries and numerous urine samples that were needed. And although 456 signed consent, 188 in the placebo run-in, only 18 finally were randomized to treatment. Electronic informed consent was used, and patients were randomized to receive tolterodine or placebo over a 12-week period. The outcome was determined using electronic diaries, and there were 2.4 less micturition events per 24 hours in the treated arm, as opposed to 0.8 micturition events less in the placebo arm. Treatment difference was 1.6, which was not significant statistically. And I think the lesson from that trial was that it was very important to focus on simplicity in decentralized trials and make sure that patients can comply with the requirements. Recognizing the potential opportunities that decentralized clinical trials offered to address challenges in trial accessibility, we published a draft guidance in May this year on decentralized clinical trials for drugs, biological products, and devices. And I'm going to spend the next few slides going through some of the important points raised in this guidance. So firstly, remote trial visits. Investigators can conduct trial visits remotely using video conferencing or other suitable technology. And this may make trial visits much more convenient for patients living far away, but clearly there are challenges. In many states, local regulations on telemedicine may make this difficult to implement. Physical examinations are not really possible when you're doing a remote trial visit, although the investigator may be able to supervise somebody doing the examination in the patient's room. Video photography may not fully capture the features of a lesion where you want to detect things like induration, tenderness. Patient engagement in the absence of in-person contact is sometimes a challenge. There are certain patients who prefer to see their providers in person rather than getting onto a telemedicine visit. Complex drug administration procedures are a challenge during a remote visit and may need specialized trial personnel on site. And finally, close medical supervision is really not suitable, for example, when one anticipates infusion reactions. And in these situations, remote visits are probably not appropriate. Direct distribution of investigational products. Well, the investigator should control the release of products to trial participants. Remember that the investigator is responsible for administering the trial drug. Local state laws differ on direct distribution to patients. Some of them may require locally licensed healthcare professionals or pharmacists to prescribe the investigational product. Packaging and handling are important considerations, particularly when we're dealing with unstable or labile medical products. And finally, plans have to be made for disposal of unused product. What about the use of local healthcare providers and local facilities? There are many examples where local healthcare providers or local practitioners perform tasks that are not directly related to the care of the patient. For example, an insurance company may get a local healthcare provider to do an insurance medical, or a local healthcare provider may do a visual acuity test for a driver's license. And in the same way, we believe that local healthcare providers are an important resource in clinical trials. These are qualified people in the clinical care environment, and they obviously bring their expertise to the clinical trial arena. Routine clinical activities can be delegated to local clinics or local healthcare providers. For example, things like x-rays, clinical examinations, and lab tests. Obviously, it's important to ensure that these individuals are appropriately qualified in the community. We mentioned in the guidance that tasks logs allow investigators to keep a record of who is doing what. We've had a lot of comments in the docket indicating that they didn't agree with this, and we will take a more careful look at whether this is necessary. Another important aspect is that the data has to be regularly reviewed by investigators, seeing they're not doing these examinations or these procedures in person. Electronic informed consent is already in widespread use. It allows patients to review and sign the consent at their homes. It may allow the provision of videos and graphics to make the process more informative and more easily understood. And of course, important to remember that signed consent must be obtained before enrollment of patients. Home visits are another novel approach. These can be done either by dedicated trial staff or by local healthcare providers. Mobile trial units are also being developed. These would be vans that travel in the community where trial-related procedures can take place. And these strategies are another way of extending the physical reach of the trial. Digital health technologies, as I mentioned before, these provide access to continuous or frequent data. They give us the ability to detect sporadic events, for example, seizures, arrhythmias, and falls. They allow patients to report their outcomes in real time, something that's often called ecological momentary assessments. They provide real-world environment evidence of drug effect, for example, while patients are working, exercising, and sleeping. And they also are able to provide an objective record of functionality. And for more information on digital health technologies, I refer to our guidance on digital health technologies published in January 2022. What about oversight? The investigator, as in any trial, is responsible for oversight of the clinical trial. The investigator and sub-investigators should be listed on Form 1572. As far as oversight of local healthcare providers, such as doctors, nurses, radiologists, delegated to do trial-related activities, investigators should review their reports regularly and make sure that there's no missing data or that data are not erroneous. And when patients present with unrelated complaints to local emergency rooms, neighborhood clinics and pharmacies, obviously these institutions are not part of the trial and not subject to investigator oversight. As far as safety goes, high-risk products and severe diseases may not be suitable for decentralized clinical trials. It would be important to ensure that patients can contact study staff if they run into a clinical problem. And it's also important to ensure that local medical facilities should be available for urgent care if the patient runs into a problem with adverse events or other trial-related issues. What about inspection? A site inspection would generally take place where the data are and where the study staff can be interviewed. As far as local healthcare providers, local inspections may be necessary if problems are found, and ancillary medical services like emergency rooms and neighborhood clinics would generally not be subjected to inspections, just as they are not for traditional clinical trials. A couple of other considerations. Not everything can be accomplished remotely. Many decentralized trials will end up as hybrids, combinations of remote visits and visits to investigator sites where procedures such as detailed physical exams and other in-person activities are required by the protocol. Supervision, as I mentioned, is important. Local healthcare providers and local facilities need to be supervised by investigators, and records need to be available from local healthcare facilities, allowing inspectors to recreate the trial. I wanted to finish off with a couple of thoughts about trials in clinical practice settings or point-of-care trials. These provide the opportunity to use existing clinical infrastructure, particularly when it's supported by interoperable data systems like electronic health records, and this has become an area of increasing interest. The recovery trial in the UK for COVID-19 was the poster trial for trials in a healthcare setting. In that trial, they reportedly recruited 40,000 COVID patients through the National Health System in the UK within the space of 6 weeks. Quite an extraordinary achievement. And they were able to show the mortality benefit of steroids in treating patients hospitalized with COVID with hypoxia. And this has now become standard practice. Practice settings allow engagement of large numbers of patients in short periods of time, and they reflect the effectiveness of treatment in real-world environments, and they provide accessibility of clinical trials to patients who wouldn't normally participate. So it leads me to my conclusions, which are that there are important opportunities to improve trial efficiencies, convenience for patients, and the ability to access diverse participants and rare diseases. Electronic technology has broadened the scope of decentralized activities considerably. Contacting FDA. If you're considering an innovative trial design for a drug-specific application, you should contact the relevant review division for a pre-IND meeting. For engagement on digital health technologies or decentralized clinical trial designs that are not related to a specific drug development program, you can contact us at the email address given here, and we will determine the best forum to address this. A couple of questions. Justification of an endpoint involves ensuring that the DHT is accurate and reliable, true or false? Well, that's false. Remember that verification and validation ensure that the DHT is accurate and reliable. Justification of an endpoint is a clinical determination and has nothing to do with the method of measurement. All local healthcare providers participating in a clinical trial would need to be listed on Form 1572, the statement of the investigator. Is that true or false? And that's false. It's just investigators and sub-investigators who need to be listed on Form 1572. And finally, which of the following is not a strategy to decentralize trial-related activities? Video and telemedicine visits, direct distribution of product, electronic informed consent, visits by participants to trial sites, and the use of local healthcare providers and facilities. And clearly, from what we've been through in the lecture, visits by participants to trial sites are what we're trying to avoid with decentralized clinical trials. So that would be a strategy that is not designed to decentralize trial-related activities. Thank you.
Hello, I'm John Concato from the Office of Medical Policy in the Center for Drug Evaluation and Research, and I'll be discussing real-world evidence. As objectives and an outline of the presentation, I'll first describe the scope of FDA's real-world evidence program. I'll move on to recognize the intersection of scientific and legal and regulatory issues related to study design in the real-world evidence era. Next, we'll interpret terms commonly used for study design and drug development, and then we'll move to identifying examples of real-world evidence in drug approvals. Let's start with the 21st Century Cures Act of 2016, which prompted FDA to establish a program to evaluate the potential use of real-world evidence to support a new indication for a drug already approved or to satisfy post-approval study requirements. A draft real-world evidence framework was issued in December of 2018, and draft guidance for industry was issued in late 2021. It's important to note that our standard for substantial evidence remains unchanged, and we can also mention that commitments were met under the Prescription Drug User Fee Act 6, and there is a new Advancing Real-World Evidence Initiatives program in PDUFA 7 that started in October of this year. The 2018 Real-World Evidence Framework applies to the Center for Drug Evaluation and Research, as well as the Center for Biologics Evaluation and Research and the Oncology Center of Excellence, but not to other centers. The program involves internal agency processes, external stakeholder engagement, demonstration projects, and guidance development, although I only have time to discuss the last two of these four components. FDA has formal real-world definitions from our 2018 framework. On the left, we see real-world data or data related to patient health status, or the delivery of healthcare routinely collected from a variety of sources. We often think of electronic health records, medical claims, registries, and the other sources shown. On the right, we see that real-world evidence is defined simply as the evidence regarding the usage and potential benefits, risks of a medical product derived from the analysis of real-world data. And note that randomized trials can generate real-world evidence when they incorporate real-world data. Let's now turn our attention to study design, starting with traditional terms used to describe patient-oriented research for several decades. On the left, we see descriptive studies, including case reports and case series. And on the right, we have cause and effect or analytic studies with a control or comparator group, which are usually described either as observational studies or what could be called experimental studies, but with an intact patient as unit of observation. For all intents and purposes, randomized trials are the most common experimental study design. When considering randomized studies to evaluate drug outcome associations, we see that we have patients at baseline who receive a drug or a comparator who are then evaluated for an outcome. When we ask the question whether the validity of the comparison is affected by sources of methodologic bias, we recognize that randomization promotes balance at baseline to help minimize bias, and for decades, randomized trials have been the preferred method for evaluating drug safety and efficacy. This second of two design slides shows non-randomized studies, where patients at baseline again receive a drug or a comparator, but this time in usual clinical practice, and an evaluation of outcome is subsequently done. Is the validity of this comparison affected by sources of methodologic bias? Well, observational studies need to address baseline imbalances to minimize bias, such as accounting for the drug of interest being given preferentially to patients who are more likely to have either better or worse outcomes. A topic related to the prior two slides is shown here. Most, if not all of you, have seen this or similar hierarchies of study design, which evolved in the 1990s, designating randomized controlled trials as a gold standard study design and suggesting other study designs are not trustworthy. More recently, however, emphasis has been placed on strengths and limitations of different study design types. One view is represented by an article titled "The Magic of Randomization versus the Myth of Real-World Evidence." The article has the quote, "Because of potential biases in observational studies, such studies cannot generally be trusted. The replacement of randomized trials with non-randomized observational analyses is a false solution to the serious problem of ensuring that patients receive treatments that are both safe and effective," close quote. I'll just mention here that few, if any, people are suggesting the replacement of randomized trials. Nonetheless, in contrast, we have a misunderstanding randomized trial article that argues, quote, "Any special status for randomized trials is unwarranted. Which method is likely to yield a good causal inference depends on what we are trying to discover, as well as on what is already known," close quote. Several CDER staff publish on this topic in an article titled "Randomized, Observational, Interventional, and Real World, What's in a Name?" The take-home points include that FDA is evaluating whether and how observational studies intended to evaluate efficacy can contribute persuasive results from scientific and regulatory perspectives. And in this context, a randomized trial versus observational study dichotomy is overly simplistic as shorthand for strength of study design to support causal inferences. Additional comments are also shown in this excerpt. To better understand the term real-world evidence, We should begin with the term big data, which first appeared in the computer science literature during the 1990s, often referring to data too large to be stored in then conventional storage systems. It's unclear when big data became the buzzword of the day, or really what it exactly means, but one perspective is that the integration and analysis of large-scale data has always been integral to epidemiology, but what's changed is that modern technology has increased the quantity and forms of available data, as well as the speed to manipulate and merge such data. If we focus on the term real-world evidence, we should recognize that real-world is actually a non-specific modifier, and real-world data and real-world evidence appeared in the medical literature as early as the 1970s or before that even, but in various contexts. In contemporary usage, however, RWD and RWE have formal regulatory definitions as I described. So one perspective is that the older epidemiologic terms were sufficient, but the emergence of big data and the enactment of 21st Century Cures Act has led to what turns out to be sometimes confusing use of different taxonomies for study design. As just one example, when you hear real-world evidence study, don't automatically think of an observational study. You really need to hear specific details to classify the study design. Based on that historical overview, we can now talk about contemporary terms for study design. An interventional study or clinical trial is where patients are assigned to treatment by a study protocol. A non-interventional or observational study is where patients receive treatment during routine clinical care. Importantly, a study can have components of both. For example, an externally controlled trial with an interventional treatment arm and a non-interventional control arm. Those same terms are used in an article from earlier this year titled Real-World Evidence, Where Are We Now? You see in the top row randomized interventional studies, non-randomized interventional studies, and non-randomized non-interventional studies. We tend, however, to jump down to the next row to talk about traditional randomized trials, trials in practice settings, externally controlled trials, and observational studies, with the main body of the figure showing different design types. It's also important to note that the generation of real-world evidence does not occur when a traditional randomized trial uses real-world data only for planning purposes on the left, such as to assess enrollment criteria or to support selection of trial sites, and that it's an increasing reliance on real-world data going left to right, and we do finally catch up with the observational study category on the far right. To clarify when real-world data generates real-world evidence, this more recent article by FDA co-authors provides even more granular details. This table is from the same article, and I won't go into all the details, but you could see that the content describes when interventional studies, aka randomized controlled trials, as well as externally controlled trials, and non-interventional studies do or do not generate real-world evidence. I'm repeating this slide that was shown earlier in the talk to pivot back to FDA's real-world evidence program. As mentioned, demonstration projects and guidance publications represent two major initiatives to promote the appropriate use of real-world data and real-world evidence. The next several slides will go into some detail on these two topics. First, demonstration, that is, research projects, represent FDA's contribution to advancing the science of real-world evidence. In informal categories of improving the use of or quality of real-world data, in the middle we have studies looking at study design proper, and on the right, so-called tools to improve real-world evidence. I'll only go across the top row for the sake of time to point out that a OneSource project with the University of California, San Francisco is designed to improve the quality of EHR data at the bedside. The next slide will show a little bit more about the RCT duplicate trial emulations, wherein real-world data combined with observational study design were used to try to emulate previously completed or ongoing randomized trials. And on the right in the top row, evaluation of confounded treatment effects was a study with the University of North Carolina that looked at the available methods to assess unmeasured confounding. This slide shows a snapshot of the primary publication from the RCT duplicate project coordinated by investigators at Brigham and Women's Hospital. I'll just emphasize the first sentence that states, quote, real-world evidence studies can reach similar conclusions as RCTs when design and measurements can be closely emulated, but this may be difficult to achieve, close quote. I refer you to the article for further details. Time also doesn't allow me to do more than list the series of FDA guidance documents on real-world data and real-world evidence. The bottom line is that we're sharing our current thinking on the topics shown, which can be grouped in categories of data considerations, submission of data, applicability of regulations, design considerations, and procedural issues. We have 2 more guidances to publish, and after receiving public comments, each of the guidances will be converted from draft to final. Let's now segue to FDA's approach to evaluating real-world evidence. Key considerations are summarized here. First, whether the real-world data are fit for use. Second, whether the study design can generate adequate scientific evidence to answer the regulatory question. And third, whether the study conduct itself meets FDA regulatory requirements. A final and more recent example is a new indication for a drug called Prograf based on real-world evidence. The drug, tacrolimus, had been approved for prophylaxis of organ rejection in patients receiving liver, later kidney and heart transplants based on traditional randomized trial evidence, and the drug was being used widely in clinical care. For various reasons, randomized trials were not done for lung transplant, but the sponsor submitted a supplemental new drug application to FDA with a non-interventional quote-unquote real-world evidence study. The data and design were evaluated according to FDA standards, and approval for preventing rejection and death in lung transplant was granted in July of 2021. Applying the considerations shown on an earlier slide to the Prograf approval, we see that the data came from the U.S. Scientific Registry of Transplant Recipients involving all lung transplants in the U.S. during the period shown. The design was a non-interventional observational treatment arm compared to historical controls. And FDA determined this study to be adequate and well-controlled, or highest evidence bar. Of note, the outcomes of organ rejection and death are virtually certain to occur without therapy, and this dramatic effective treatment helped to preclude bias as an explanation of results. I don't wish to leave the impression that working with real-world data to generate real-world evidence is easy. In fact, this slide highlights what can and often does go wrong in the same 3 categories of data sources, study design and analysis, as well as study conduct. To highlight just a few, data reliability and clinical relevance are often lacking in the first category. In the second category, confounding has been mentioned before, as well as complicated problems with the index date or zero time that I don't have the opportunity to discuss in detail. And then last but not least, under the conduct of randomized studies, the protocol and analysis should be pre-specified, and FDA requires access to patient-level data and the ability to inspect real-world data sources. In summary, FDA's real-world evidence program is advancing as outlined in the agency's 2018 framework for real-world evidence. It's important to recognize that new terminology linked to the emergence of big data and the passage of 21st century cures is often used inconsistently, and randomized trials versus observational studies is an oversimplified dichotomy. It's also true that older terms for study design in drug development are now joined by newer terms describing those same study designs. But regardless, FDA approves drugs and biological products using real-world evidence based on applicable and existing regulations, maintaining our evidentiary standards. Finally, a knowledge check. True or false, randomized trials are not within the scope of real-world data, real-world evidence at FDA. This statement is false. Randomized trials generate real-world evidence when they incorporate meaningful real-world data. The second statement, real-world evidence studies for effectiveness are held to a different, that is, lower evidentiary standard than randomized trials. This statement is also false. Regardless of the type of study design, the FDA has specific standards for judging whether a product is safe and effective for use. Thank you very much for your attention, and I look forward to the question and answer discussion period.
Fred, Leonard, and John, thank you all so much for excellent presentations. The good news is we have a very engaged audience. We've gotten several questions in the chat, so we'll see how many we can get through over the next 15 minutes or so. Hopefully you're all able to join me with the audio for this chat. It looks like we've gotten questions for several of you. Why don't we start with Fred? So Fred, one question we have, how can an investigator ensure enrollment of the right subject, with right in quotes? That is a subject who's not too sick so as to render treatment ineffective, or not too well so as to decrease the probability of reaching an endpoint.
Can you hear me? This is Fred.
Yes, Fred, go ahead.
Okay, and should I turn my camera on or leave it off?
Off is fine, thanks, Fred.
Okay, that's not an easy task to enroll the right patient. What you need to do is, during the investigator meeting or when you are reviewing a protocol, look at the inclusion and exclusion criteria. If you start seeing class, like for example in cardiology, New York Heart Association class 1 or 2, these are patients who could pretty much act normally, and that would give you a clue that this may not be the right patient to see a treatment effect unless there's a huge sample size or there's a trajectory that leads to expeditious degradation of the health status. And on the other hand, if you see a lot of class 4 subjects that are includable or included in the clinical trial, that's a clue that they're too sick. So look very carefully at the exclusion criteria. And when you enroll a patient, try to be patient, meaning try not to enroll a patient because you need to have 2 or 3 patients per month to meet your quota, and accidentally enroll patients that might fall outside of the not too sick and not too well population. So this is a careful scrutiny of clinical presentation, your evaluation of the patient status when you're enrolling them, making sure that they're just the right patient. That's easier said than done.
Thanks, Fred. And you have another question here. How can an investigator know whether or not the results of a trial may be regression to the mean? How does an investigator guard against this when the contribution to the overall sample size is small at a specific site?
Yes, that's a very good question. And how one would guess, one would never know, because you're enrolled in a patient and you're in isolation. You're at one site, one investigator, one sub-investigator, one patient comes in, and you have to have a good clinical sense as to what the average presentation is in this patient population, what they can and cannot do. And when you gauge the mean status of the patient, and then you look at the patient in front of you that you're about to enroll, are they looking like it's too well? Are they too well, or are they too sick relative to what you feel is the mean? This gives you a sense that at baseline, if you're enrolled in a very sick patient at baseline, they might get better because of concomitant medications or because of the natural history of variability of the disease. So again, look at whether or not your patient that's in front of you represents the tail end of a mean presentation, and that might give you a clue as to the potential risk for regression to the mean.
Great, thank you. And Leonard, we have questions for you on both DHTs and DCTs. Why don't we start with a couple on DHTs? So the first, does digital health technology have to be approved for medical use to be used as a digital health technology in a clinical trial? The example they give is an Apple EKG. Or is validation of the technology for the investigator's purpose allowed for the clinical trial? For instance, comparison of a self-written app to a gold standard.
Thanks, Kim. It's not a requirement that every DHT has to be cleared or approved. If a DHT that's used in the clinical trial is being used as a device, as a medical device, then it does have to be cleared, but otherwise not. So, for example, things like activity trackers and so on do not have to be cleared by CDRH, but they clearly have to be validated to make sure that they're measuring the clinical feature accurately.
Great. And then maybe a follow-up question. How about a cell phone to record EKGs?
I think there's no problem with the use of a cell phone, provided the technology has been validated to make an accurate recording. So again, it all depends on the validation. That would tell us that the measurement made by a cell phone is equivalent to a measurement that might be made with a traditional machine. And we also have encouraged the opportunity to use, for patients to be able to use their own devices, to bring their own devices, provided the devices sort of satisfy the same specifications and can do the same thing. So yes, cell phones probably going to play increasing roles in decentralized, or at least digital health technology measurements. Thanks.
And one more in this category. What is the FDA stance on the use of, quote, bring your own device, for instance, cell phones, for the ECOA PRO data collection? There is quite a bit of evidence on the ability for this modality to collect patient data accurately. How much more data would be needed for the FDA to support this? Experience of myself and colleagues' experience indicate that there's some reluctance.
So again, I touched on that in the previous answer, but I think FDA is definitely interested in facilitating that. I think we all understand that provided devices that are owned by study participants may be very effective in capturing the data if they all satisfy the minimal specifications, depending on what's needed. So for general things like patient-reported outcomes and so on, where the demands on the technology are limited, it's very likely that bringing your own devices will be satisfactory, but obviously that would have to be reviewed. For more specialized kind of measurements, like things like pulse oximetry or glucose measurements, I think there'd probably be a little bit more circumspection about whether bringing your own device is a good idea. We would have to make sure that all the devices provided equally reliable information. But I think we all understand that nobody wants to run around carrying dozens of cell phones in their pockets or being strapped to 6 smartwatches. So I think in the interest of convenience, it's important to try and facilitate the fact that patients are able to use their own devices, which they're familiar with and comfortable operating. Thanks.
Great, thank you, Leonard. John, a couple of questions for you here as well. The first, I was taught that observational studies are inferior to clinical trials. So why should we even bother with real-world evidence?
Thanks, Kim, and thanks for the question. I think the question refers likely to hierarchies of study design, which I mentioned in the presentation. They gained traction as of the 1990s. Unfortunately, when applied rigidly, those design hierarchies oversimplify the question of whether any particular study does generate valid evidence. We all agree that randomization is wonderful, and I'll add that the prospective research infrastructure that accompanies a trial is another underappreciated strength. But on the other hand, small sample sizes and other issues can and sometimes does threaten the validity of trial results. If we switch gears, I could say conversely, at the start, bias and confounding are indeed serious threats to non-interventional studies, but such studies can be valid. As I mentioned in the presentation, we use a 3-pronged approach to get started. First, are the data fit for use, sometimes called fit for purpose? Is the study design adequate, i.e. rigorous? And last but not least, third, whether the study conduct is appropriate and adheres to our FDA regulations. As an aside, as was mentioned in the example of tacrolimus Prograf, when the anticipated effect size based on available knowledge suggests that the drug effect is large, that helps us to believe that the drug outcome association is real, even if we can't identify and account for every particular type of bias. So the bottom line is we still need clinical trials, make no mistake, but we shouldn't summarily dismiss observational, aka non-interventional studies. Thank you.
Thanks, John. And one more for you while you're here. In what way can randomized trials involve real-world data?
Thanks again. I did mention it perhaps briefly in the talk. As one example, electronic health records as a real-world data source can be used when appropriate, say to obtain information on the primary outcome in a point-of-care randomized trial. Both I and Leonard Sachs mentioned that particular type of study design. And I should add, in addition, most trials, if not all, already sometimes use data from EHRs, albeit in a modest way, say to incorporate laboratory values or other information from the quote-unquote bedside. But that practice, sometimes called the swivel chair activity, is where someone transfers, on the trial staff, transfers information from the records to the case report form, but that sort of flies off below the radar screen. So what we're talking about when randomized trials generate real-world evidence is more than, say, identifying patients for a trial or identifying sites or capturing their CBC, their complete blood count, or their chemistries, but rather have a fundamental impact, such as collecting the outcome for a trial. In that case, including our congressionally mandated need to report back to Congress how real-world evidence is being used in drug development. We're keeping tabs on both non-interventional, that is, observational studies, as well as clinical trials, sometimes generating real-world evidence. So I'll stop there, Kim. Thank you.
Great, thanks, John. Fred, a couple more questions for you. The first is, I did not understand the interest in evaluating the intent to treat, or ITT, population. Why evaluate participants that did not receive treatment?
The reason why you want to evaluate patients who did not receive treatment is when you randomize a patient, you need the results of all the patients that were randomized. For those who did not receive treatment, there might have been a reason why they did not receive it. They might have dropped out of the study. They might have been intolerant to the patient, to them when they took the drug or study drug. There might have been a number of reasons, but to preserve the integrity of the randomized process, you want to not potentially bias the patient population by beginning to exclude patients that might cause a bias. So that is why we're interested in the ITT population. We've modified that a little bit. We tolerate the modified intention to treat by all patients who received at least one dose of drug. And you could ask the same question. If they only received one dose, that might not be effective at all. But again, we're doing our best to preserve the integrity of the randomization process. If you go to the other extreme, just those patients who participated in the trial fully, those who did not violate the protocol, where you don't know why people violated the protocol, there could have been safety concerns or lack of efficacy, then you have a restricted population that is fully biased, that is independent of the randomization procedure that you had wanted to install in the first place. So that is why we are very interested in an ITT population. The minute you randomize them to some arm, you stick to it.
Great, thank you, Fred. Leonard, another question for you, this time on decentralized trials. How is the FDA adapting its regulatory framework to ensure the integrity and reliability of data collected through decentralized trials, especially considering the diverse range of digital health technologies and remote monitoring tools used in these studies?
There are no specific changes to our regulations, at least at this time. A lot of these features have already been part of trials for many years, as I mentioned in my presentation. A lot of decentralized activities have taken place before. Some of the digital health technologies are also no different from technologies that have been used in traditional trials, things like ECG machines and blood pressure machines and glucose machines and so on. So technologically, I don't think we're in a completely new universe. There are a couple of considerations for data, where we've expanded on some of the regulations. Many of the folks on the call may be familiar with our Part 11 regulations, and we have a guidance published, and now it's being revised and will be issued in final pretty soon, on the considerations for data integrity and reliability as far as the electronic processing of the data goes. I think that's a novelty. Again, I don't think that there are any other fundamental differences between whether something's done locally or remotely, provided the data are supervised and the investigator knows what's going on. So the short answer is there are no new regulations, but obviously we've had to interpret older regulations in the context of these more novel trial designs.
Great. Maybe one additional follow-up question there that's related. In a decentralized trial, what sort of training is necessary for local healthcare providers?
I'm glad you asked that question, because our vision of local healthcare providers is that they should not need training. The reason we're interested in including local healthcare providers in trials is that we're using them for what they normally do, and they should not need to have a knowledge of the trial protocol or the investigational product. For example, if you're getting a local healthcare investigator close to a patient's home to do an ECG or to do a physical examination, that would be agnostic to the fact that the patient was on a particular investigational drug. For all those procedures that require knowledge of the protocol, for example, study-specific assessments or scoring of outcomes and so on, that should be done by study staff. We do not envisage local healthcare providers having to do that. That would require training, obviously. So the short answer is that there should not, based on the tasks that are allocated to local healthcare providers, training should not be required, although there's a possibility they may be given limited instructions, like have a patient sit for 5 minutes before you measure the blood pressure, but no specific training. Thanks.
Great, and thank you all. We have a lot of excellent questions, and really appreciate all of your input on this session and the work you did to prepare for it. With that, I believe we are up to our first break, and we will resume at 1:30 p.m. Eastern Time. Thank you all.